Primary databases. Primary nucleotide sequence repository. The main databases that store and provide the raw nucleic acid sequences are Genbank, EMBL and DDBJ. Genbank is located at USA and is accessible through NCBI portal. EMBL or European Molecular Biology Laboratory is located at UK and DDBJ or DNA Data Bank of Japan is located at Japan. All the three have uniform data formats but not identical and exchange information in a daily basis. All these databases are the part of International Nucleotide Sequence Database collaboration. As all the three have same data format, Genbank database can be taken as an example to explain the data entry. Genbank entry is in the form of a flat file. Simply say the file contains the extension of .gbk. GBK files are plain text files that are best viewed in a fixed width font. It is easily parsed by computer programs because the fields containing different types of information are well labeled. The header of the file contains information describing the sequence such as its type, shape, length and source. Features of the genome sequence follow the header and include protein translations. The DNA sequence is the last element of the file which ends with a double slash. Here is an example for a Genbank format. Here is a Genbank flat file format obtained from mus musculus DNA containing the cytosine 5-methyltransferase 3A mRNA. The source is mus musculus, which is a house mouse, and here you can see the cloning and characterization of a family of novel mammalian DNA methyltransferases. In this image, you can see the various fields such as locus, definition, accession, keywords, organism, etc., which would be studied in the next table. Now, we will see the various fields which are obtained from a Genbank entry and we would study in detail about each field. Locus. The locus field contains a number of different data elements including locus name, sequence length, molecule type, Genbank division and modification date. The locus name in this example is AF068625. The locus name was originally designed to help group entries with similar sequences. The first three characters usually designated the organism. The fourth and fifth characters were used to show other group designations such as gene product for segmented entries. The last character was one of a series of sequential integers. Next, we are going to study about sequence length. It is the number of nucleotide base pairs or amino acid residues in the sequence record. In this example, the sequence length is 200 base pairs. Molecule type. The type of molecule that was sequenced in this example is mRNA. Genbank division. The Genbank division to which a record belongs is indicated with a 3-letter abbreviation. In this example, Genbank division is ROD. The Genbank database is divided into 18 divisions such as PRI for primate sequences, ROD for rodent sequences, MAM for other mammalian sequences, VRT for other vertebrate sequences, INV for invertebrate sequences, PLN for plant, fungal and algal sequences, BCT for bacterial sequences, VRL for viral sequences, PHG for bacteriophage sequences, SYN for synthetic sequences, UNA for unannotated sequences, EST for EST sequences, PAT for patent sequences, STS for sequence tagged sites, GSS for genome survey sequences, HTG for high throughput genomic sequences, HTC for unfinished high throughput cDNA sequencing and ENV for environmental sampling sequences. The next is modification date. It is the date in the locus field of the last modification. The sample record shown here was last modified on 6th December 1999. The next field is definition. Definition describes the brief description of sequence, which includes information such as source organism, gene name or protein name, or some other description of the sequence function. If the sequence has a coding region, description may be followed by a completeness qualifier such as complete CDS. The next field is accession. Accession is a unique identifier for a sequence record. An accession number applies to the complete record and is usually a combination of letters and numbers, such as single letter followed by 5 digits, for example U12345, or 2 letters followed by 6 digits, for example AF123456. Some accessions might be longer depending on the type of sequence record. The next field is version. Version describes a nucleotide sequence identification number that represents a single specific sequence in the Genbank database. This identification number uses the accession. Version format implemented by Genbank or EMBL or DDBJ in February 1999. The next field is GI, also known as the Gen Info Identifier Sequence Identification Number. In this case, it is 6449467. If a sequence changes in any way, a new GI number will be assigned. Keywords. Keywords describe a word or a phrase describing the sequence. If no keywords are included in the entry, the field contains only a period. The next field is the source. Source describes the free format information including an abbreviated form of the organism name, sometimes followed by a molecule type. Organism. The formal scientific name for the source organism, the genus, species, and its lineage based on the phylogenetic classification scheme from the NCBI taxonomy database. Reference. Reference describes all the publications by the authors of the sequence that discuss the data reported in the record. References are automatically sorted within the record based on date of publication, showing the oldest references first. Authors. List of authors in the order in which they appear in the cited article. Title. Title of the published work or tentative title of an unpublished work. Journal. Medline abbreviation of the journal name. PubMed or PubMed identifier. Direct submission. Contact information of the submitter such as institute or department and postal address. This is always the last citation in the references field. Some older records do not contain the direct submission reference. However, it is required in all new records. Features. This describes the information about genes and gene products as well as regions of biological significance reported in the sequence. These can include regions of the sequence that code for proteins and RNA molecules as well as a number of other features. Source. Source describes the mandatory feature in each record that summarizes the length of the sequence, scientific name of the source organism and taxon ID number. It can also include other information such as map location, strain, clone, tissue type, etc. if provided by the submitter. The next field is taxon. Taxon is a stable unique identification number for the taxon of the source organism. A taxonomy ID number is assigned to each taxon in the NCBI taxonomy database. The next field is CDS or coding sequences. Coding sequences are regions of nucleotides that corresponds with the sequence of amino acids in a protein which includes the start and stop codons. Base 1 to 200. Base span of the biological feature indicated to the left. Features can be complete, partial on the 5' end, partial on the 3' end and or on the complementary strand. The next field is protein ID, which describes a protein sequence identification number similar to the version number of a nucleotide sequence. Protein IDs consist of 3 letters followed by 5 digits, a dot and a version number. The next field is translation. The amino acid translation corresponding to the nucleotide coding sequence or CDS. In many cases, the translations are conceptual. Gene. This is a region of biological interest identified as a gene and for which a name has been assigned. The next field is complement. This indicates the feature that is located on the complementary strand. The next field is origin. The origin may be left blank, may appear as unreported. This information is present only in older records and is given in FASTA format. Primary protein sequence repositories. The two important protein sequence repositories are PIR-PSD or Protein Information Resource Protein Sequence Database at the National Biomedical Research Foundation, USA, and Swissprot at the Swiss Biotechnology Institute, Switzerland. PIR-PSD. PIR is a resource to assist researchers in the identification and interpretation of protein sequence information and was established in 1984 by the National Biomedical Research Foundation or NBRF. PIR-PSD is a collaboration of PIR, the Munich Information Center for Protein Sequence, Germany, and the Japan International Protein Information Database, Japan. The superfamily based classification of protein sequence is one of the unique characteristics of this database. PIR-PSD also has the homology domain and the sequence motifs based classification. Homology domains correspond to the evolutionary building blocks and sequence motif represents the functional sites or conserved regions. PIR-PSD is available online at pir.georgetown.edu. Swissprot. Swissprot is an annotated protein sequence database created at the Department of Medical Biochemistry of the University of Geneva and is a collaborative effort of the department and the European Molecular Biology Laboratory (EMBL) since 1987. The data entry can be considered as core data and annotated data. Core data is a sequence entered in the single letter amino acid code, related references and bibliography. The taxonomy of the organism from which the sequence was obtained is also part of this core data. The annotated data section contains the functions of the protein, post-translational modifications like phosphorylation, acetylation, etc., functional and structural domains and sites like calcium binding regions, zinc fingers, secondary structure features like alpha helix, beta sheet, similarities to other proteins if any, and diseases related with that particular protein. Swissprot is available online at www.uniprot.org. The entry in Swissprot and the explanation have been described in the table. Let us now study the various codes which are used in the Swissprot entry. The first column gives us the code, the second column gives us the expansion, and the third is the explanation. The first is ID. ID helps us in identification. It gives the unique name for the entry and also the information about the entry. AC is the accession number. This is the stable way of identifying the entry. The AC number will not change for an entry. If an entry has more than one AC, shows that the entry is generated by merging multiple entries. DT gives us the date. We can see 3 different dates for an entry. Those are the creation date, the modification date of the sequence and the annotation date respectively. DE gives us the description. It gives the general description about the sequence. GN is the gene name. It gives the name of the gene that codes for the protein. OS is the organism name. It is the name and taxonomy of the organism. OG is organelle, information regarding the organelle that contains the gene. OC is organism classification. It gives us information about the classification of the organism. RN, RP, RA, RX, RT and RL gives us the reference number, position, cross-reference, comments, authors, title and location. Bibliographic representation of the sequence. It also gives us the information on the extent of work carried out by the authors. CC gives us the comments, text document that contains extra information about the entry. DR is the database cross-reference, gives cross-reference with other database which contains information regarding the same entry. KW is the keywords. It gives keywords that can be used as indices to find required information. FT, features table. This describes the regions or sites of interest in the sequence, for example, modifications, binding sites, etc. SQ is the sequence header. It indicates the beginning of the sequence data and gives a brief summary. Uniprot KB, Swissprot TREMBL, Uniref, Unipark are different types of Swissprot database. Uniprot KB. Uniprot knowledge base is the collection of functional information on proteins with accurate, consistent and detailed annotation. This includes widely accepted biological ontologies, classifications and cross-references and clear indications of the quality of annotation in the form of evidence attribution of experimental and computational data. It consists of two sections. One is containing information extracted from the literature and curator evaluated computer analysis with manually annotated records, and the second section contains the computationally analyzed records with awaiting manual annotation. These two sections are referred as Uniprot KB or Swissprot, which has been reviewed, manually annotated, and Uniprot KB or TREMBL, which is unreviewed and automatically annotated. Uniprac. Uniprot archive is a comprehensive and non-redundant database that contains most of the publicly available sequences. Uniprac avoided redundancy by storing each sequence only once with a unique identifier. Unipark has combined many databases into one at the sequence level, and searching Unipark is equivalent to searching many databases simultaneously. Uniref. Uniprot reference clusters contains clustered set of sequences from Uniprot KB and selected sequences from Uniprac to obtain the complete coverage of the sequence. The sequence fragments are merged in Uniref before saving. Both PIR, PSD and Swissprot use user-friendly software that helps the user to get only the required information. Swissprot also use a sequence retrieval system or SRS that helps to search through other relevant databases like TREMBL. TREMBL released as a supplement to Swissprot and is a computer annotated protein sequence database. It contains the translated sequence entry for all of the coding sequence present in EMBL nucleotide database which has not been fully annotated.